Hi everyone, my name is MehrAzin, and in this presentation I will be talking about the contribution of growth hormone and insulin-like growth factor 1 to glucose metabolism and insulin sensitivity. This presentation is based on a relatively recent research paper, and throughout this presentation I will be going over the objectives, the overview, results, and conclusions of that research. The objective of this study is to assess the direct and indirect effects of growth hormone on glucose metabolism in peripheral tissues at the molecular level, and to discuss results from human studies investigating the effect of growth hormone administration on glycemic control and insulin resistance. So in my previous presentation regarding biochemistry and society, I have discussed growth hormone deficiency, growth hormone treatment, and recombinant growth hormone. In that presentation, I've also went over the importance of IGF-1 growth hormone axis. And I will review that one more time because it will be important for the further discussions. So under normal conditions, growth hormone is produced within the somatotropic cells of the anterior pituitary gland, which is then secreted by the pituitary into the circulation. The secretion of growth hormone is controlled by two different hormones that are secreted by the hypothalamus. Growth hormone releasing hormone promotes the secretion of growth hormone, and somatostatin inhibits the secretion of growth hormone. Growth hormone secretion occurs in a pulsable manner, and it contributes to increased levels of IGF-1 or insulin-like growth factor, which is made mainly within the liver, but it can also be made within other target tissues, including the growth plate. Insulin-like growth factor 1 or IGF-1 plays a pivotal role in mediating the actions of growth hormone. So this will be quite important in what is going to be discussed throughout this presentation. Insulin-like growth factor 1 is commonly known as IGF-1, which is a single-chain polypeptide of 70 amino acid residues cross-linked by 3 disulfide bridges. IGF-1 displays obvious homology to proinsulin. Proinsulin is the pro-hormone precursor to insulin made in the beta cells of the islets of Langerhans within the specialized regions of the pancreas. In humans, proinsulin is encoded by the INS gene. The number of differences in amino acid positions between IGF-1 and insulin suggests that duplication of the gene of the common ancestor of proinsulin and IGF occurred before the time of appearance of the vertebrates. Moreover, IGF-1 also has metabolic effects similar to insulin in modulating glucose metabolism. So what are the effects of growth hormone on glucose metabolism? Growth hormone is important for promotion of somatic growth and the regulation of substrate metabolism. Metabolic action of growth hormone occurs in multiple tissues, including the liver, muscle, fat, and pancreas, either directly or indirectly through insulin-like growth factor 1 or IGF-1. The diabetogenic action of growth hormone has been well described in previous in vivo studies. Studies investigating the effects of growth hormone on glucose metabolism have demonstrated that growth hormone increases glucose production through gluconeogenesis and glycogenolysis from the liver and kidney. Thus, growth hormone has been established as one of the counter-regulatory hormones against insulin action, considering that like glucagon and epinephrine, growth hormone helps prevent hypoglycemia by increasing gluconeogenesis and glycogenolysis and decreasing glucose uptake and consumption from peripheral tissues during fasting. The diabetogenic effect of growth hormone was supported by the high prevalence of diabetes in acromegaly patients. This role of growth hormone has been further supported by Bernardo Jose, who was an endocrine physiologist who found that hypersensitivity to hypoglycemic effects of insulin was increased in hypophysectomized animals, and insulin sensitivity was reduced after the administration of anterior pituitary extract. So for the sake of time, I will not be able to go into much detail about the fascinating work of Bernardo Jose, which made great changes in understanding the pathophysiology of carbohydrate metabolism and the mechanism of action of certain diabetic conditions. But it's provided the link to the article about his work and his contribution to endocrine studies. And you guys are more than welcome, if you're interested, to go ahead and read more about Jose's work. It is also important to note that nutrition influences growth hormone secretion, and it varies between fasting and fed states. Hypoglycemia is when the blood glucose levels are too low, causes acute raise in growth hormone secretion that is dependent on both the severity and the rate of glucose fall. On the other hand, hyperglycemia, which is when the blood glucose levels are too high, causes the suppression of growth hormone secretion. So as you can tell by now, growth hormone, insulin-like growth factor 1, and insulin levels in the blood are associated with each other. Another important thing to understand is that growth hormone and the feedback loops that are associated with growth hormone have influence on glucose metabolism and insulin sensitivity. Naturally occurring growth hormone is secreted from the anterior pituitary and excretes its effect through stimulation of the growth hormone receptor. Growth hormone also has other potent effects, including modulation in carbohydrate, fat, and protein metabolism, promotion of lipolysis, increase in amino acid uptake and protein synthesis, and decrease in protein breakdown. The growth promoting and other anabolic effects of growth hormone are mediated through stimulating the production of insulin-like growth factor 1 or IGF-1, which was something that I discussed in more details in the previous presentation when I talked about the growth hormone and IGF-1 axis. The effect of growth hormone on systemic glycemic control is complex, partially due to its indirect effects via insulin-like growth factor 1, which has glucose lowering effects similar to insulin. So as I've mentioned in the previous slides, insulin-like growth factor 1 demonstrates similar homology to different types of insulins and to the pro-insulin hormone. Here's an example where adults with growth hormone deficiency are paradoxically associated with abdominal obesity and insulin resistance, which may be partially associated with their reduced IGF-1 action. So here is another reference slide to the previous presentation where I discussed the synthesis of recombinant human growth hormone, or IGH. In this slide, I will briefly go over recombinant human growth hormone, because in the upcoming slides, I will be discussing the effects of human growth hormone therapy on glucose metabolism and insulin sensitivity. So structurally, recombinant human growth hormone is a single domain, non-glycosylated globular protein containing 191 amino acids, and it has the same amino acid sequence as the naturally occurring human growth hormone. So in addition to its utilization for the treatment of growth hormone deficiency, recombinant human growth hormone can be used for a wide range of reasons and to treat a wide range of health conditions. In addition, recombinant human growth hormone can not only promote linear growth, but also improve body composition and metabolism, increase bone density, and improve the quality of life, which are the characteristics that I mentioned in the previous presentation in regards to bodybuilders and bodybuilders utilizing growth hormone treatment, or the topic about the utilization of growth hormone treatment in elderly patients in terms of improving longevity. So recombinant human growth hormone is indicated for long-term treatment of pediatric patients who have growth failure due to an inadequate secretion of endogenous growth hormone, and for long-term replacement therapy in adults with growth hormone deficiency of either childhood or adult onset, which are also what we've discussed in the previous presentation. The serum insulin-like growth factor 1, or IGF-1, is a good biochemical marker of recombinant human growth hormone action, and it is often used to evaluate the efficiency of RHGH preparations, which has some limitations, but it is a commonly used compound for testing the efficiency of RHGH. So in this slide, I will be discussing growth hormone therapy and insulin sensitivity. Growth hormone therapy is known to antagonize insulin's action on peripheral tissues such as the skeletal muscle, liver, and adipose tissue, thereby increases glucose production from the skeletal muscle and liver and decreases glucose uptake from adipose tissue. A number of human studies have suggested that growth hormone administration in growth hormone deficient adults may reduce visceral adiposity and improve cardiometabolic disturbance. Meanwhile, IGF-1 that represents similar characteristics to insulin actions in the skeletal muscle and liver and increases the circulating IGF-1, increases in the circulating IGF-1 after growth hormone administration, may have beneficial effects on glucose homeostasis and insulin resistance. And insulin production is increased to compensate the increased circulating glucose after growth hormone administration. So based on these outcomes that have been observed and recorded from the different studies that has been done assessing the effects of growth hormone therapy on carbohydrate metabolism and insulin sensitivity, we can conclude that growth hormone, IGF-1, and insulin sensitivity are definitely correlated. The clinical presentations of growth hormone deficient adults are characterized by increased visceral adiposity, insulin resistance, dyslipidemia, and hyperglycemia, which contributes to increased risk of cardiovascular morbidity and mortality. And as I've mentioned, a number of human studies have suggested that growth hormone administration in growth hormone deficient adults may reduce visceral adiposity and improve cardiometabolic disturbance. And growth hormone-induced lipolysis or lipolysis in the visceral adipose tissue and subsequent increased circulating free fatty acids also interferes with insulin signaling pathways, and chronic exposure to free fatty acids may exert direct toxicity in beta cells. So the overall outcomes of this study demonstrated that different studies in children and adolescents also suggested that growth hormone administration may induce insulin resistance in short-term treatment, but its long-term consequences have not been fully determined. Some other studies raised concern over increased insulin resistance and impaired fasting glucose during growth hormone treatment, especially in patients with obesity and in elderly patients. International cohort studies indicate that growth hormone therapy may increase the incidence of type 2 diabetes in children and adolescents with predisposing risk factors. Therefore, it is prudent to monitor possible negative consequences on glucose metabolism during and after growth hormone administration. And finally, large-scale longitudinal cohort studies are required to examine the long-term influence of growth hormone therapy on cardiovascular outcomes in growth hormone deficient children with or without continuation of growth hormone after cessation of skeletal growth. Thank you so much, everyone, for tuning in for this presentation. If you guys have any questions, I would be more than happy to answer them in the discussion board section of Blackboard.